So first thing first, let's draw the entire ovarian cycle, right? So here we will have like a hormone concentration, 0 to 100. And here we will have length of the ovarian cycle. So day 1, and what's the maximum?
28.
Very good, 28. Okay, so typically the ovarian cycle is about 28 days. How do you start counting the ovarian cycle? What happens on day 1?
The first day of menstruation.
Very good. Menstruation should be happening. So as soon as you get your period, you start counting, right? So this is going to be day 1. What happens on day 14?
Ovulation.
Ovulation. Ovulation is when the follicle ruptures, right? So as you remember, we have different types of follicles: primary, secondary, tertiary, and Graafian. So which one will be rupturing in the ovulation?
Graafian.
Okay, so the major difference between tertiary and Graafian follicle, as you remember, it's just that Graafian follicle is super big. So if we go to the previous slide, so this picture, I'm always confusing because I have one picture. Okay, it is, no, it's not there. Okay, because I was confused because I have one picture in embryo and one picture in histo, so I always mix them up. Okay, so here the first phase, this phase, this is known as the follicular phase. So if I go back, right, so the first half of your ovarian cycle, you will call this phase follicular. This is the follicular phase. Guess what happens during the follicular phase?
The follicle is...
The follicles are growing, right? So that's what we have seen before. Primary, secondary, tertiary, Graafian. Cool. The next one, this is going to be the luteal phase. Okay, so here from day 14 to day 28, this is known as the luteal phase. And in the luteal phase, you're going to see that something is going to be quite dominant, and this is actually corpus luteum, right? So corpus luteum is going to be dominant. So let's talk about the 2 hormones that we have here. This picture is good. It's good. Okay, so day 1, okay, day 14, day 28 that we saw here as well. This picture is good because you can see that there's a bit of the rise in LH, a bit of the rise in FSH, but this is a mistake here a bit because FSH is higher than LH at first. So if I go back to my slide here, here will be FSH. Okay, this is the FSH, which is rising. And below it, a bit lower concentration is the LH, which is rising as well. Could you tell me which 2 hormones, which hormone is responsible for this rise in FSH and LH?
GnRH.
GnRH, okay?
GnRH.
Releasing hormone, so hypothalamus. So hypothalamus is going to be more, you give more the echo, more gold stuff. You're giving me the echo, sorry. Okay, so yeah, so you're gonna see that hypothalamus is going to give you a boost, right? It's gonna give you a boost here on day 1. GnRH, FSH, LH starts to rise. Of course, you can guess that during the follicular phase of the ovarian cycle, which hormone should be higher, FSH or LH?
FSH.
Of course, because all of these follicles are going to be growing, they need stimulation, so you're going to see that FSH will always be a bit higher. But something will be happening at this area right here. The closer you approach the ovulation, something will be happening here. As you can see, if we look at another hormones, we'll also see that there are 2 other hormones that we're also very interested in, estrogen and progesterone. Could you remind me, how do we make estrogen?
The granulosa cell.
The granulosa cell.
So let's do the whole process. You remember that corpus, not corpus, in the follicles that we see here, right? These follicles, we have their granulosa cells like that. And on the top, we have the theca interna cells. So can we do the whole process? Theca interna cells have
Receptor.
for what?
For
LH. LH, okay? So theca interna will have LH receptor. LH will be binding to theca interna cells. Theca interna cells will make weak estrogen, right? So they'll make a precursor of the estrogen. And who will take up this precursor of the estrogen?
The granulosa.
Granulosa cells. And granulosa cells will convert this precursor of the estrogen to the normal estrogen. And you remember, how do we call the enzyme that we do this with? It's called aromatase. Okay, so aromatase. So granulosa cells possess aromatase, and they will convert this estrogen precursor into the normal estrogen. So which hormone kind of stimulates the production of estrogen? LH, right? Because LH binds and we make the estrogen. So you're going to see that here LH rises, right? So it starts to rise here. And FSH here does the opposite. It drops just a bit. Why does the FSH drop before the ovulation? So why would we see here that FSH is dropping like this? Just a bit, but it's dropping. Why would it be dropping? This is the negative feedback system in a way, because what is the follicle stimulation hormone doing? It's actively stimulating follicles to grow. I'm about to ovulate. My follicle is about to rupture, right? So do I need so much stimulation? Of course not. All the other follicles are soon going to become dead. So FSH is not needed anymore. So through the estrogen, FSH is going to be going down. So the reason why FSH kind of drops here is because we have estrogen, because your granulosa cells are actively making estrogen here. But why are they actively making estrogen? Because LH slowly starts to rise. Why is the LH slowly rising? Why is it actually going just a bit upwards? Because approximately on day 13, hypothalamus gives you the second boost. So there are 2 times in the cycle, hypothalamus is super active. So approximately 1 day before the menstruation and approximately 1 day before the ovulation. So is it correct to say that hypothalamus secretes GnRH in a continuous manner?
No.
This is a trick question, because if you're an embryo, then it's yes. If you're an adult, then it's a no. Okay, so I love 3 questions, so there you go. So if you're an adult, hypothalamus does not secrete GnRH in a continuous manner. It secretes it in a cyclic manner, right? So a bit 1 day before menstruation, a bit 1 day before the ovulation. But if you're an embryo, GnRH is secreted continuously, because you need to differentiate to become whatever gender you are, male or female. And LH, FSH constantly need to be high. So on day 13, we have a second boost of GnRH. And as you can guess, what's going to be happening? Your FSH will rise just a bit. It will rise just a bit because GnRH. But LH, you're going to see, it was constant, constant, constant. But then when there is the second boost here of GnRH, LH does something like this. And this has a name. It reaches almost 100% concentration. This is called LH surge, right? So there is a sudden rise of LH. The question is why? Why would FSH just rise a bit, but LH would rise a lot? I always like to give this particular analogy because it's quite useful to remember it for the rest of your life. If you imagine that LH is fire, estrogen is like oil or like, I don't know, benzene for the fire. If you add just a bit of it, right, the fire will just go ballistic, will just blow up completely. So why does LH actually surge? Because estrogen is there, because estrogen passes its threshold accumulation, do you see? So we have so much estrogen that there is actually a reaction between estrogen and LH. So in order for you to have the LH surge, your estrogen should pass the threshold, it should pass its normal concentration. You should have a lot of it. What is going to be happening because of the LH? Because of the LH, you have ovulation. Because of the LH surge, you have ovulation. Why do you have ovulation? Because of the LH surge. You remember that here we had antrum. Remember, in the follicles we had antrum. Inside of the antrum, there was a fluid, right? We called it liquor or follicular fluid, something like that. Who makes this follicular fluid? Granulosa cells. So the cells which are outside of the antrum, granulosa cells, they're making this fluid. But why do they do so? Because of LH. Because LH binds to granulosa cells, and granulosa cells will make this fluid. So can you imagine what will happen if I suddenly have too much LH?
Too much.
The rupture.
Too much fluid in the follicle. Follicle cannot withhold that much fluid. It ruptures, it bursts. And this is what we call
Ovulation.
ovulation. That's what you see here, right? So your follicle has ruptured, it bursted, that's it. So here, again, this picture is not good. I don't know why I always choose shitty pictures, but okay. Okay, so here we have the LH, it's quite high, LH surge, and here on this picture is showing you as this LH just suddenly goes down. It's not like that, it's actually a bit like that. Gradually, it starts to return to its normal concentration like that, very gradual. It takes about 9 days for your LH to reach its normal normal concentration, okay? So 9 days it takes. When LH is super high, inside of your follicle, as you remember, we have remnant cells, right? We have the theca interna and granulosa cells. So this is what is left in the follicle after ovulation. Because LH is super high, you're going to see that these granulosa cells and theca interna, which are left behind, they undergo a process which is called luteinization. And luteinization basically means that these cells turn to become some kind of endocrine cells. Okay, so they get some lipid droplets into themselves, and they use these lipid droplets to produce some kind of hormones. Because it's luteinization, the process, because they need high LH in order to undergo luteinization, these cells are called lutein cells. So the remnant cells in the follicle, granulosa and theca interna cells, due to high LH, undergo a process called luteinization. During luteinization, after luteinization, they become lutein cells. And lutein cells make two hormones, progesterone and estrogen. 75% of its secretion is progesterone, and 25% of its secretion is estrogen. Okay, so majority of the secretion of corpus luteum is progesterone. That's why you can see that progesterone is super high, right? So it's going up, 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 up. So in the second phase, in the luteal phase, the dominant hormone is progesterone. But in the follicular phase, the dominant hormone is estrogen. So what happens next? As you can understand, LH comes down because it cannot be high forever. So eventually it just comes down to its normal concentration. This amount of LH is not enough for the lutein cells to be alive. Okay, so they arose when it was super high LH. Okay, super high. So you still need super high LH to be alive. So if LH comes down to an end, right, if it comes down to its normal concentration, lutein cells will undergo apoptosis. Okay, so no more LH to feed them, or it is there, but it's not enough of the LH to feed them. So you're going to see that they undergo apoptosis. That corpus luteum, as you know, is called
Corpus albicans.
It's going to be known as the corpus albicans, right? So that corpus luteum will be called corpus albicans, and that's what we see here. That's the case if fertilization does not happen. By the way, when you have corpus albicans, what happens to your progesterone? Anybody makes it?
No.
So it drops, and what is that going to be doing?
You shed
The period.
Forgot the name.
We shred the endometrium, right? So we are going to be shredding the inner lining of the uterus. We shred it, but why? It's very important to remember that inside of the endometrium, we have spiral arteries. And these spiral arteries are there to keep your endometrium thick, because you can see during the estrogen phase, we were making it thick, we were proliferating the endometrium to keep it thick, right? But progesterone is the one that keeps it thick. So estrogen makes endometrium thick, but progesterone keeps it thick. How does it keep it thick? It's super thick, so it needs lots of nutrients to be alive. So who provides those nutrients? Spiral arteries, they're right here, right? So these are spiral arteries, they're right there. Just one problem, these spiral arteries are super progesterone sensitive. If progesterone is present, spiral arteries are open. If progesterone is absent, spiral arteries are closed. They constrict. Now imagine, no embryo. Corpus luteum dies. Nobody makes progesterone. Progesterone drops. What happens to spiral arteries? They contract, they constrict. Endometrium is not receiving blood. No nutrients. So what's going to happen to endometrium? It starts to die. As it dies, it starts to peel off. And that's what we call is menstruation. So do you understand what's the role of progesterone in endometrium? So if I have an embryo, what should I have at a constant rate? Progesterone. Progesterone should be constant. So can embryo bring the LH back?
No.
So don't forget, LH is controlled by hypothalamus. We have two phases, right? So phasic secretion by the hypothalamus, by GnRH and all of this stuff. So definitely embryo cannot control that. So embryo will make a hormone, as you know. This hormone is known as the HCG, human chorionic gonadotropin. It is a gonadotropin, so it looks exactly as LH. So somewhere at this point, HCG will be rising if you have an embryo. For your lutein cells, it doesn't matter. LH, HCG, they are identical. Pepsi Cola doesn't matter. Okay, so we will take anything of those things. And corpus luteum continues to produce progesterone. The corpus luteum, which is in the pregnancy, is called corpus luteum graviditalis, right? Pregnant corpus luteum. And remember that normal corpus luteum is under the influence of LH, but corpus luteum graviditalis is not under the influence of LH. It's under the influence of HCG, because the embryo makes HCG and keeps it constant. Okay. Okay, so here I did not finish, but you get the idea. Okay, so it's like this. Okay, FSH is also going to be like normal, normal, normal. Okay, but yeah. Okay, that's what everything I have said is just here. So few terms before we continue. So capacitation and fertilization. As you remember, capacitation, we remove the acrosome region. It says here protective barriers, but you know what I mean, right? So acrosome is being removed of the spermatozoa, because behind the acrosome we have the enzymes which are needed to digest the zona pellucida, right? So we need to expose it in the capacitation. Before fertilization happens, these digestive enzymes need to attach to zona pellucida, and it triggers acrosome reaction. But don't forget that these enzymes are actually inactive. So something needs to activate them. What activates them? ZP3, right? So as you remember, your zona pellucida has three proteins there, ZP1, ZP2, ZP3. And ZP3 is the one which is going to be activating the digestive enzymes there. Once you penetrate the zona pellucida, once you digest it, you're going to penetrate it. Only the head of spermatozoa squeezes in, and the two pronuclei will fuse, right? So we have male pronucleus, female pronucleus. That's just the nuclear of the oocyte and spermatozoa. They're going to fuse, and zygote will be arising, right? So fertilized oocyte, this is the zygote. Inside of the zygote, you have either XX or XY chromosome. It's a one cell, but you will already have inside of it XX or XY. Zygote will be undergoing the cleavage. As you remember, that's when we actively divide the cell. 2 cell, 4, then 8, then 16, and blah, blah, blah. So here it's quite important to know in histology that zona pellucida will be changing its consistency after fertilization to prevent polyspermy, because we already got one spermatozoa, so now we don't want to have others, so we're going to try our best and not allow other spermatozoas to enter as well, because if too many spermatozoas enter, then I don't have 2 chromosomes, I'm going to have like 3 or 4 or something else, right? So super weird thing will be there. So how can I prevent polyspermy? 3 ways are possible. Fast block to polyspermy is when your plasma membrane of the oocyte kind of creates an electrical shock. There is like a electrical part goes there, so all spermatozoas get blocked by this electricity. It's very, very short, okay, relatively short because it's 1 minute, but then can you imagine for oocyte to have its plasma membrane depolarized for 1 minute? That's long for oocyte, but for us it's of course short. So that's one way. Just depolarize the membrane, all of the spermatozoa will not want to come in. Next thing is the cortical reaction. You can just change the polarity of the oolemma, okay? So of the plasma membrane of the oocyte has something to do with calcium, sure. And zonal reaction, okay? So you're going to see that the rest of the zona pellucida will not be digested. Kind of it keeps the way it is, and on the opposite, it even creates more proteins there by cross-linking them. So you're kind of building up a thicker wall. So that's why it is kind of correct to say that after fertilization happens, zona pellucida becomes just a bit thicker, a bit rougher, stronger, because you add a bit more proteins there. So those are the 3 ways to prevent polyspermy. Okay, so that's what you will do there. Here we're basically talking about the corpus luteum, that it's under the influence of paracrine and endocrine secretions. So paracrine are the hormones which are going to be, or hormone-like substances which are made by the ovary themselves, right? So ovary will make it and ovary will be affected. That's paracrine secretion. So that's the estrogen and insulin growth factor. Insulin growth factor will also be made by ovaries, and endocrine parts as well. That's not made by the ovary, but it will affect your ovary. So for example, HCG, right? LH, prolactin, and insulin will also be affecting you here as well. We have said that corpus luteum makes progesterone until when? Maximum until the end of the 3rd month, so until the end of the 1st trimester. That's when the corpus luteum actually makes progesterone. But already starting from the end of the 2nd month, placenta starts to chip in and it makes progesterone as well. So by the end of the 3rd month, corpus luteum will be degenerating, it will be breaking down, it will be disappearing, but it's okay because placenta is already making all of that progesterone. So fallopian tube can be divided into 4 areas. So we'll have isthmus, ampulla, infundibulum, and fimbria here. The epithelium inside is pseudostratified, columnar, ciliated, and some cells are non-ciliated. That cells are called PEG cells. So epithelium is quite interesting here because some cells do have cilia, some cells do not, and these cells that do not have cilia are called PEG cells. The cells vary in height according to the hormone that, like, remember we just talked about. So if I go back, we have the estrogen phase and we have progesterone phase. So it depends under which hormone you are right now. So we will see it right now. Okay, so all the ciliated cells they swipe the oocyte towards the uterus. PEG cells, the non-ciliated cells, are actually secretory cells. They secrete nutrient-rich secretion for one reason. If you actually have an embryo, so we can kind of provided with the nutrients, right? Estrogen is going to stimulate cellulogenesis. Progesterone increases number of secretory cells. So if I go back, right, to this picture here, during the estrogen phase in the epithelium of the fallopian tube, which cells are dominant? Ciliated or PEG cells?
Ciliated?
Ciliated. Why are ciliated cells dominant here? Because if you have an oocyte, you need to swipe it. You need to swipe it towards the uterus, making sure that it's going to be definitely getting closer to the spermatozoa just a bit. But in the progesterone phase, the PEG cells are going to be quite active. They make this nutrient, right, rich fluid for the reason if you actually have an embryo. So maybe we'll need to supply this embryo with some kind of nutrients. Layers, right, mucosa, muscularis layer will consist of the inner circular, out longitudinal, and then serosa is outside. Okay, so here is just the same thing that we have talked about, nothing interesting, that actually because fallopian tube has this muscle layer, it can actually do peristalsis. So you will see that sometimes it will be like a wave of muscle contraction, which will be literally squeezing your oocyte closer to the oocyte like that, to the uterus like that. Okay, so it just pushes your oocyte closer to this area as well. So that's your slide right here. This is going to be the uterine tube, right, fallopian tube. This is the mesosalpinx. And inside of the mesosalpinx, you see this cyst-like structure. This could be either apophoron or paraphoron. Do you know what it is? No. We'll talk about it in embryo.
It's a remnant of the no epididymis. That
We talked about previously, remember? There are
efferent,
two
no?
ducts. In embryo, there are two ducts. There's Mullerian duct and
Graph and Wolffian.
Wolffian, very good. Mullerian duct will become uterus and fallopian tubes. Wolffian duct will become vas epididymis, vas deferens, and a bit of a prostate as well. So as a female, do I want to keep the Wolffian duct? Of course not, so I need to get rid of. But I cannot get rid of it completely. There are some kind of remnants of it, and these remnants are known as the apo and para or foron. So this one is one of them. Because of the sections we cannot know, okay, but this is either apo or para or foron. Okay, remnant of the Wolffian duct. How do you call remnant of the Mullerian duct in male? It's called appendix of testis. On the testis, there is a bit of protrusion there. They call it appendix of testis. So that's the fallopian tube here. You can see this is the mucosa, right? And this is the muscularis layer. If you take a look at the mucosa, you can see that there are lots and lots of ciliated cells. Do you see? Lots of them have cilia, and like just a couple of them do not have cilia, like here, PEG cells. This is also PEG cell. Do you see? No cilia, right? So judging from this picture, which hormone is right now dominant?
Estrogen.
Estrogen. Okay, so this is taken in the half of the cycle. So estrogen is dominant right now, right? So you can see that also because of estrogen, the mucosal folds are not as long. They're quite short. So because of estrogen, these mucosal folds are not as long. This is the muscularis layer, right? You can see it's quite vascular structures here, lots and lots of blood vessels. And then this is just serosa, for example. Another slide for you, also fallopian tube. So this is the mesosalpinx, the same idea, but you can see we cannot see the apo or paraphoron. These are probably like lymphatic vessels. They look like that. We need to magnify a bit, but it looks like a lymphatic vessel. This is also a lymphatic vessel, also, right? So some arteries are here, right? But yeah. So notice that the length of the mucosa definitely has increased. If we compare with the previous slide, like this one, and here, mucosa is highly folded and super tall. And if we magnify, we see lots of PEG cells. all of these cells are PEG cells. There are lots of them. There are still some ciliated cells, but majority of the cells are PEG cells. If you see so many PEG cells, you know that the hormone which is dominant right now is progesterone, right? So it's going to be progesterone. So these PEG cells are super into secretion. Okay, so the same idea. So which cells are these?
Ciliated.
Ciliated. These cells are
Peg.
PEG cells. Yeah, you can see on the slide they're even a bit darker, which also helps. Like this is also a PEG cell. Okay, what are these? What is this line?
Basal bodies.
Very
Support.
good. Yes, basal bodies. So don't forget that under the cilia we have the basal bodies, which is just the M talk.
Julia,
Ah,
on the surface, it's myocytes. Like, oh no, it's
on the surface. Where on the surface?
no. Here. This picture. Yeah,
This one?
yeah.
This is not on the
Ah,
surface.
no, it's a basement membrane. No,
It's
I'm
a
looking
basement
upside.
membrane.
Yeah. Sorry.
It's a basement membrane because that's the surface. Okay,
That's the
so
empty
these
column.
cells are sitting on this part. So we have a bit of the basement membrane, but this is also in a way lamina propria, like this part here. This is lamina propria. Basement membrane is just here. It's just this area. Okay, so this is actually lamina propria with like fibroblasts and stuff.
Okay.
Yeah. So uterus, hollow, pear-shaped organ, pelvis between bladder and rectum, sure. And the female that have never given birth, it's very tiny, about 30 to 40 grams. All the diameters are here, right? Anatomically, uterus has body, okay, that's the major part there, and cervix, it's the neck of the uterus. From the cervix, vagina continues. The uterine wall consists of 3 layers. We have the endomyo and perimetrium. Endometrium is the epithelium. It's the one that is constantly proliferating and then being peeled off every month. Myometrium is the muscular layer, so it has a bunch of smooth muscle cells there. We'll see that the way the smooth muscle cells run in the myometrium is like 8. That's how they run. Okay, so smooth muscle cells run like that. So there's a bunch of smooth muscle cells that are connecting to each other, and that's the way that they run. So when the uterus kind of contracts, it legit squeezes. Like it squeezes it. When the uterus contracts, can you imagine this area contracts, it squeezes whatever is present in the uterus. Perimetrium is just serosa, just a fancy way of saying that's serosa. So it's just connective tissue which is present on the surface. Of course, I don't need to tell you that endometrium, myometrium will be constantly undergoing cyclic changes each month. So menstruation and all of this stuff. So myometrium, it has 3 indistinctly defined layers. So there will be 3 layers: supravasculare, vasculare, and subvasculare. Those are the 3 layers of myometrium. I'll show them on the slide, but basically, which one is closer to the endometrium? Subvasculare. Which one is closer to the perimetrium, the outer? It's going to be supravasculare. Okay, so that's the most important part. They're here talking about just some additional information, like the way they're running, but as I have said, the most important thing, that kind of like an 8 shape. Endometrium, more important. So endometrium has 2 layers, basale and functionale. Functionale is further subdivided into 2, spongiosum, and then the most superficial is compactum. So stratum compactum is actually facing the lumen of the uterus, right? So if we do the layers, this is the lumen of the uterus. So first one is stratum compactum, then stratum spongiosum. Together, they are known as the stratum functionale. Next thing is going to be the stratum basale. In the stratum basale, you will have the stem cells. So during menstruation, which layers will be disappearing?
Compactum.
Only? It's actually the entire functionale, that's what it's called. It's the functional layer, right? So compactum and spongiosum will be disappearing, leaving the basale only. So leaving only the stem cells. And then again, the stem cells proliferate, and then again gets peeled off. So functionally, the entire thing will be actually peeling off during the menstruation. And under the stratum basale, you'll have the myometrium. Myometrium will have the 3 layers as well, right? So vascular layer will be here, and above it, the supravascular, and below it is the subvascular, right? And then the most outer layer is the perimetrium. Within the endometrium, we have uterine glands. They are super, super deep. They're super big, okay? They're humongous glands, and they contain glycogen. They are lined by the simple columnar epithelium, and inside of them we'll have like a mixture of the secretory and ciliated cells as well. But we'll see that there's like kind of very, very interesting glands. So for example, here, this is the uterine gland, right? So as you can see, it's within the entire endometrium. So that's the basale, that's the functionale. Don't forget the top one. This is going to be the compacte, and this is the spongiosum here, right? So the 2 sublayers of the functionale. In this particular slide, we need to talk about the blood supply of the uterus, okay? So blood supply of the uterus, because here it's very interesting how the blood runs. So take a look, we're inside of the myometrium, we're in the vascular layer. So that's why it's called vascular layer, because it actually possesses the big branches of the uterine gland, right? Uterine gland gives you bigger branch as well, which is the arcuate artery, the same as in the kidneys we saw them, right? So arcuate arteries, but here it's a different type of arcuate. And then you're going to see that arcuate arteries gives you a radial branch. So radial branch is within the subvascular layer of the myometrium, right? So don't forget, this is the vascular layer. This is going to be the supravascular layer, right? And this is the subvascular layer. Radial branch is the one that is kind of entering into the stratum basale. And in the stratum basale, you're going to see that it kind of gives you 2 branches. So radial artery will give you 2 branches here. It will give you straight and it will give you spiral. And if straight artery only stays within the basale, right, doesn't go forward to the functionale, the spiral is the one that starts to kind of push forward into the stratum functionale. And then you're going to see that eventually this spiral artery, because it is an artery, right, it becomes arterioles, and then these arterioles are continuing to kind of become capillaries and etc. The capillaries in the endometrium, specifically in the compact layer of endometrium, will be very special. So capillaries here are quite special because they actually possess like a dilation of some sort. So here the capillaries are super special. They possess an additional dilation. And this additional dilation is called lacuna. You can think about lacunas as lakes of blood, like additional lakes of blood. And these lacunas are quite important for just general vascularization of endometrium, if the embryo comes in or like something like that. So you just have additional pools of blood that you're going to be calling lacuna. So lacunas are dilated ends of the capillaries. Now, which one is going to be peeling off, right? So this entire area, so stratum functionale, all of it peels off during the menstruation. So you can see that proximal part of the spiral artery remains, right? So only the distal part of the spiral artery could also disappear with the menstruation. So point of this slide here, remember the blood supply and remember this. Don't forget that spiral artery is very sensitive to
Progesterone.
to the level of progesterone. Okay, so menstrual cycle, we have talked about it, right? So cycle we just talked about. So what is happening? Basically, very important to remember that endometrium will actually have 3 phases. So first is the menstrual phase. So first couple of days of the first half of the ovarian cycle, right? As you remember, the first half is called follicular phase, right? And then we call it the luteal phase, but that was for the ovary, right? That's what we talked about. What about endometrium? So the follicular phase of the endometrium is actually 2. It's menstrual and proliferative. So first couple of days is menstruation and then proliferation. So menstruation proliferation is under effect of which hormone?
Estrogen.
Estrogen. Okay, so you will see that in the beginning, the estrogen will be low because nobody is making it, right? So follicles are just starting to grow. But then closer to the end of the proliferative stage, estrogen will be super high. And then luteal phase of the ovary is equivalent with the secretory phase of the endometrium, which is regulated by the progesterone, right? So here it will be progesterone, which we talked about in the beginning of the class. Estrogen makes the endometrium thick, but progesterone keeps it thick. Make sure that it keeps on secreting something. Okay, so yeah. So let's talk about it. Uterus proliferative stage, right? So when is it exactly? 5 to 14th day of the cycle, because the first couple of days is the menstruational stage. Facilitated by the FSH or specifically by the estrogen, right? So estrogen is responsible for this stage. That's why in your self-control test, you will see that the question will be something like this. Proliferative stage of the uterus is which hormone dependent? It is estrogen dependent. Okay, so this another name for it is estrogen dependent. You remember the uterine glands, right? So they're within the entire part of your endometrium, within the entire part of the stratum functionale and basale. Very important to remember that these uterine glands in the estrogen or proliferative stage of the uterus are straight. That's how, by simply looking at the endometrium, I can tell you which phase are we at right now. And you can see that they're straight. Do you see? They're straight. The shape that they have, they're straight. If you see that these uterine glands are straight, you know this is proliferative stage. You know that this is estrogen stage. You know that the endometrium is right now repairing itself after menstruation, right? Compactum, spongiosum, we talked about it. In the secretory phase, take a look. These are also your uterine glands. Are they straight? Absolutely no. The shape that they have is actually corkscrew. Everybody knows what is a corkscrew?
The one,
The wine thingy, yeah, that's what I also say. That's how I detect who is into the wine thingy. High five. Okay, so I can tell you, I can detect all of these corkscrews like that. Okay, so yeah, it's the wine thingy. You know, like when you open the wine, it's like this particular
wine opener. Yeah.
Exactly, the wine opener. Exactly. So that's the corkscrew. That's what it is basically. But yeah, okay. So the wine opener is like it has the spiral appearance. So if you look at the uterine glands, you can see that these are the spiral or corkscrew appearance of the uterine gland. So definitely this is the secretory stage of the endometrium, which is regulated by progesterone, right? So it's progesterone.
Julia, the definition of a corkscrew or all the shapes are like are good to say in the exam?
Yeah. Yeah, you need to say them, yeah. Because like, yeah, they will ask you what's the shape.
And it's like to say
Corkscrew, yeah.
Corkscrew.
Yeah. That's if you open the book, that's what is recommended saying, yeah.
Okay.
You can say spiral, okay? You can say corkscrew. It's up to you. But like from experience, they like when you come up with those weird things to describe it, you know? So like they're like, because again, it kind of shows that you definitely do understand what you're talking about, but you haven't just memorized like from, I don't know, lectures that it is spiral shape or something like this. But yeah, you may or you may not. I hope you do, because you'll see that the mark will go up, like from experience, the mark goes up.
Okay.
Okay.
No, nothing.
Okay. Okay, so we have talked about this part. There's just additional information, but just read it home. And the menstrual stage, right? So the menstrual stage is day 1 to 4. Of course, every girl has it different, right? So some has less, some have more, but we talked about it. So basically you have the progesterone sensitive spiral arteries, the entire reason why you menstruate, which layers are peeling off, right? So stratum functionale basically peels off, stratum basale stays, right? But yeah. Very important to know how much do you lose. 35 to 50 milliliters. In clinical practice, nobody loses like this. Okay, everybody loses, majority of people lose more. So it's definitely more than 50 milliliters. So 50 milliliters is what, like a shot, like a big shot, right? So
Less than a shot.
So yeah, like, I mean, the taller you are, the fatter you are, the more you will definitely lose. Yeah, but this number, like I'm always saying that whatever we learn in the theory years, majority of the things are useless by the time you come to the clinical practice, because all the numbers, everything is different. Okay, so in clinical practice, actually, if you have this number, it's not good. Okay, that there's something wrong. Okay, that there is less blood that you're making, like you're not under hormonal stimulation as you should. But yeah, okay, so just 35 to 50 milliliters, that's the normal according to anatomy department. Okay, so that's your uterus estrogen phase. So if it is estrogen phase, what's another name for it?
Proliferative phase.
Proliferative, yes. So it's going to be proliferative phase. So you can see that that's the endometrium, right? So that's the myometrium, and perimetrium is not visible. That's the lumen of the uterus. So immediately from this magnification, you can see that endometrium is very, very thin. So it's definitely estrogen phase. It's definitely proliferative stage because it's so thin. So that means that you have just menstruated and now your endometrium is trying to repair itself, right? So we magnify, so this is the endometrium. You can see that these structures are uterine glands and they are more or less straight, right? So because I can see that they are straight, it kind of proves to me that this is actually estrogen phase or proliferative phase. So here we'll have compactum. Okay, so these things are a bit wrong. So this is the stratum compactum. This is going to be the stratum spongiosum until here. Okay, so approximately this is stratum spongiosum. It means that this from here, okay, until here, this is going to be the stratum functionale, right? And then this area right here, that's the basale. Okay, so this part right here, that's the stratum basale. That's where the stem cells should be. Okay, this is where myometrium is already happening, but how would you call this layer approximately? I know that there is no exact layer appearance, but still, how would you call this layer in myometrium? The one closest to the endometrium.
Endometrium is the sub. It's the sub.
Okay, so this is the subvasculare. Okay, so this is where the subvasculare will be. Here within the endometrium, we can see some kind of weird looking blood vessels. So these are actually spiral arteries. Okay, so there's so many of them because they're technically one spiral artery, which is just kind of going at it like that, right? So this is the myometrium. I kind of divided it for you, right? So this is the endometrium, then maybe this one will be the subvasculare, vasculare, and then the supravasculare. Within the vasculare, that's where we're supposed to have branches of the uterine arteries. So like here, for example, these are the branches of the uterine artery. And then they go like this, right? So radial, then arcuate, right? Then the straight, then the spiral, and etc. This is progesterone phase or secretory phase of the endometrium, and you can see how much of endometrium there is, right? So there's lots and lots of endometrium present here. So even with this magnification, you can say that this is definitely not proliferative stage. This is definitely secretory stage. And you can see even with this modification, this stuff. Even with this modification, you can see that it's kind of spirally or corkscrew appearance, right? So if I see the shape like this of the uterine glands, then that's it. That's my progesterone phase. That's my secretory phase. So this is a proof for that. Again, spiral arteries, right? And these are actually lacunas, these ones, these pools of blood. Okay, so these are actually lacunas. So the same, basale, functionale. Top layer is the compactum, and this is the spongiosum. Uterine glands are visible here. You can see these cells are lymphocytes. There are lots of lymphocytes here. And these cells are stromal cells. So in the tissue of the endometrium, in addition to the uterine gland, you can see there are lots of cells here, right? So these cells here. So not the ones which are lying in the uterine gland, but the ones which are here. These cells are stromal cells. Stromal cells will be called decidual cells if you're pregnant. So if you get pregnant, they become something else. They become decidual cells. So this is why if you're pregnant, endometrium changes its name. It's no longer called endometrium, it's called decidua because it possesses decidual cells. So these cells are stromal cells. In normal endometrium, no function, okay? So just maintaining stroma of the endometrium, just sitting there, nothing interesting. But as soon as you get pregnant, they will definitely become decidual cells. And decidual cells will have lots of functions. And notice how many lymphocytes, lots of them. Okay, so same idea, just how much of the endometrium there is. So it definitely shows you that this is progesterone phase, corkscrew appearance, spiral, whatever. Vagina, fibromuscular tube extending from the cervix all the way to the vestibule. As you remember, vestibule, that's just the space between the labia minora, right? In virgins, the opening of the vagina could be surrounded by a fold of the mucous membrane called hymen. Some girls are born without hymen. It's normal, okay? So some girls during first sexual intercourse, it hurts, but they don't bleed as much, like, I don't know, 3-4 drops of blood, that's it. Some girls lose their hymen to a bicycle or to something like this, like gymnastics, if you were doing something like that, jumping, okay, too much skipping, you will lose the hymen to that. So that's why it's very interesting topic. For example, I know for a fact that in Iran and in the part of Russia where I'm from, we have something called hymenoplasty, where they give you a new hymen. Basically, you go around, pardon my French, whore around, okay, do whatever you want to do. And then when it's time to get married, okay, you go get yourself a new hymen, hymenoplasty, $2,000, and that's it, okay? Your husband will never know. Doesn't matter your past, nothing matters, because to him you will be a virgin. It's ridiculous, but that's what's in my part of Russia where I come from. That's like go-to kind of surgery. But yeah. Inner mucosal layer, so of course, because it is a tube, is going to consist of the same thing. So we have mucosa, right, the muscularis layer, and then the outer part there as well. It has numerous folds. Here they're also called ruga. So the same as in the stomach, right? So here these folds are going to be also called ruga. And the epithelium that they have is stratified squamous non-keratinized epithelium. Could you remind me who else has this kind of epithelium?
The part in the vocal cord.
Esophagus.
Esophagus. There you will also see non-keratinized, but not the same as here, because take a look. It looks as esophagus. Do you remember? This kind of epithelium, super thick, it was identical to esophagus. Remember we talked about it, because they always ask you this question. How do you know that's not esophagus? That's the question. How do you know that that's specifically vagina? Because the epithelium is the same. 2 layers, okay, quickly, and adventitia is there, the same. So how do you know that this is actually esophagus and not vagina? So first let's do layers. So this is the mucosa, then we have the muscularis layer, right? And then outside of it is the adventitia. So if you kind of magnify into the epithelium here, right? So that's your esophagus. Anything weird?
The basement membrane.
Mm-mm.
No, it's not supposed to be thick.
It's in trachea, thick. It's
Right.
urinary bladder and ureter, it's thick, but not in the esophagus. Epithelium specifically. Anything weird? Anything catches your eye? You're finishing.
The
Huh?
papilla.
Say what? Something was saying something?
The papilla.
It's a bit pale. Yeah, I agree with you. It's super pale. So why could it be pale? Why would it be pale?
Because it contains
Glycogen, right? So because
Glycogen.
it contains glycogen. So because this area contains lots of glycogen. Does your esophagus contain lots of glycogen? No. Okay, so the epithelial cells there do not have the glycogen. So the reason why this area is actually pale, because as we have just said, it contains lots of glycogen. So what would you use this glycogen for? Why would there be glycogen in the cells of vaginal epithelium? Maybe you know.
lubrication, maybe?
For
too
the
sugary.
sperms.
For the
And
sperms.
Oh, you guys are so kind. No, it's not for the sperms. No. It's definitely not for the sperms. Nobody cares about the sperms. How do they reach and what do they do? But no, it's for bacteria. Inside of the vagina, we have a very interesting bacteria. And this bacteria is super important because it makes sure that the acidity is very low. So this bacteria will be using this glycogen and making lactic acid in return. So glycogen is for something, it's for bacteria. Okay, so for bacteria which are inside of the vagina, they're good, normal bacteria, we should have them, so no need to get rid of them. They're part of the normal flora, right, of your body. They use the glycogen here, and they are going to be converting this glycogen into the lactic acid. And lactic acid is responsible for the low pH in the vagina. Okay, so just some basement membrane and a bit of the lamina propria is present here. As you can see, lamina propria is quite interesting. It has lots and lots of collagen fibers, and not only collagen fibers. If you magnify a bit, have I? No. If you kind of try to see, okay, you will see that there are lots of pink lines like this. And these pink lines are actually elastic fibers. You can see them here. So the lamina propria of the vagina is very special because it doesn't only possess collagen fibers, it will have also lots and lots of elastic fibers there as well. So now we start the fun part. We start the embryology. So I told you to refresh your things a bit, right? So you remember this. Okay, so during pregnancy, the endometrium will be called decidua, and there are going to be different parts of the decidua. like basalis, capsularis, and parietalis. So let's go to the next picture. So there you go. So here we have our embryo, right? So this is, sorry, so this is the embryo right here. This is the amniotic cavity where the embryo is chilling. And this is going to be the chorionic cavity, right? As you can see, everything else which is in like weird yellow color, this is endometrium, because embryo kind of inserts itself into this endometrium, right? You can see that it is surrounded by it. So this endometrium is on the top of the embryo as well, because embryo inserts itself into that endometrium. So this particular endometrium, which is on the top of the embryo, this is known as the decidua capsularis, because it's like a capsule, right? So this area right here, that part of the endometrium which is outside of the embryo, this is the decidua capsularis. That part of the endometrium that has nothing to do with embryo, so it is on the opposite side, this is known as the decidua parietalis, right? On the opposite side here, this is the decidua parietalis. This one, for example, will also be decidua parietalis because it has nothing to do with embryo, right? And that part of the decidua, where it gives you the blood, this is called decidua basalis. So 3 deciduas, right? So basalis, capsularis, and everything else is parietalis. Which part do you think is in the placenta?
Basalis.
Basalis. Okay, so in the placenta, you will see that there will be basalis. Okay, so basalis is part of the placenta right here. Now, in addition, you will see that there will be some kind of, how do you say it? Some kind of embryological corresponding parts as well. So these are maternal membranes. That's how the question goes. What are the maternal membranes? Basalis, capsularis, parietalis. But what are the fetal membranes? The fetal membranes are called chorion. And this part is the area where villi will be. Do you guys remember what are villi? That's the area where fetal blood vessels are located. Okay, fetal blood vessels are here. So this is known as the chorion frondosum. So here specifically, this is the chorion frondosum because here it possesses lots and lots of villi. And the part outside where there is no placenta, do I need the villi? Well, of course not. There is no placenta, so I'm not receiving anything. So here the villi will degenerate. And this area is actually called chorion leve. So this area is the chorion leve. So could you tell me what is on the top of the chorion leve?
The decidua capsularis.
Very good. Decidua capsularis. How do you call, like decidua capsularis, is it maternal or is it fetal?
Maternal.
Maternal. What about chorion leve? Is it maternal or is it fetal?
Fetal.
Fetal. Okay, so this is fetal. It's the embryonic area here. Chorion leve, is it smooth or is it bushy?
Smooth.
It's smooth. Okay, so it's going to be smooth. What about this area right here? This is chorion frondosum. Is it smooth or is it bushy?
Bushy.
It's bushy. Okay, so it's very, very bushy. Yeah. Okay, so it has like this tree-like appearance. So what is on the top of the chorion frondosum?
Villi.
Decidua basalis.
Villi are in the chorion frondosum, but on the top of the chorion frondosum, you have
Decidua basalis.
Capsularis.
Basalis. Capsularis is on the top of the liver. It's on the opposite end. So this is the chorion frondosum, this is the chorion liver. So how do you call this area together?
Placenta.
Placenta.
That's placenta. So this is a question in the self-control test. What does placenta consist of? What does it consist of?
Decidua basalis and chorion frondosum.
Decidua basalis and chorion frondosum. Cool. So here we talked about it, that stromal cells of the endometrium will become decidual cells, right? cleavage, you remember, right? So we started with the zygote, then it became 2-cell stage, then 4, right? Then 8, and then morula. Morula is the one that enters into the uterus. How do you call 4 and 8?
Blastomere.
Very good. You call it blastomere. Yes, this is going to be known as the blastomere. And then morula is here. Morula becomes blastocyst. What does blastocyst consist of? What is this?
Inner cell mass.
Another name?
Embryoblast.
Another name?
Proper.
Yes. Embryo proper. Okay, so inner cell mass, embryoblast or embryo proper. How do you call this one? Outer cell mass
Trophoblast.
or trophoblast. And what is this blue thing? This blue thing is also here, but then as you see, slowly in blastocyst, it starts to disappear. Like here, there's no more blue thing here. It's zona pellucida. So slowly it's being washed away, as you remember, right? So slowly we're gonna be removing it. Okay. This picture should be familiar from last semester. So this blastocyst is how old?
9.
Amazing. This is actually 9 days old. Very good. So this is syncytial trophoblast, cytotrophoblast. How do you call this thing together?
Bigerminal disc.
Bilaminar germ disc.
Bilaminar.
That consists. You remember the 9 days, but you don't remember the bilaminar germ disc. It's hilarious.
I remember the bi.
That's good. Okay, so epiblast, hypoblast, right? And you remember this is the Hürses membrane that came from the hypoblast. Okay, it actually does not matter any of those things. The only things that matters to us actually is this one, the syncytial and cytotrophoblast, right? Because here is the process of implantation, just for you to kind of refresh your memory that syncytial and cytotrophoblast are the ones which are, as you can see, kind of protruding into the endometrium, right? So they're kind of forcing themselves into the endometrium. This blastocyst is already implanted in the endometrium, and you can see that we have formed a new cavity called chorionic cavity, right? Outside of the chorionic cavity, we see 3 membranes. And these 3 membranes are giving you something which is called chorion. And this chorion has 3 membranes. So the most outer is the syncytial, then cyto, right? And how do you call this one? The extraembryonic somatopleuric mesoderm, but where did you get it from?
From the house.
From the Hürses, very good.
Hürses
So
membrane.
don't forget this Hürses membrane right here is intact for now, but later it kind of splits. It gives you 2, the somatopleuric and the splanchnopleuриc mesoderms, and the space between the 2, that's the chorionic cavity, right? So chorionic cavity will be between the 2. Will you have chorionic cavity when you're born? Will you have this cavity?
No, yes.
No, yes, maybe. No, you will not have it because amniotic cavity will expand everywhere. When the embryo will rotate, when it will fold, you will see that amniotic cavity will take majority of this area. Okay, so you can see that at one area, somewhere here, we have bushy appearance. There are lots and lots of protrusions here. And on this area, it's more or less smooth. So how do you call this chorion, the smooth one? Okay, go back. How do we call the smooth chorion?
Capsularis.
Live. Capsularis is the maternal. How do you call the smooth chorion? You call it the chorion live, this one, right? And how do you call the bushy one?
The chorion frontosum.
Very good. So these areas that you can see on this picture are here. So the top one is the chorion frondosum. You can see it has lots of protrusions here. These protrusions are called villi. We'll talk about them in a sec. And then at the bottom, you can see that there aren't any protrusions, right? So these are just your some kind of nothing. So it's just syncytio, cyto, and somatopleuric mesoderm. That's it. So this is the chorion leve. So placenta, we have talked about it. Two things are needed: basalis from the mama, right, and chorion frondosum from the embryo. So, okay. Okay, so let's talk about this way. So this part here, this is going to be the chorion frondosum. So this area, this is the chorion frondosum. Could you tell me what are the layers of the chorion? So what is this?
Somatopleurer.
Somatopleuric is this one.
Ah. So splanchno.
Splanchno is covered in the yolk sac somewhere here. What's the top layer of embryo? So what's the top layer?
In situ trophoblast.
Syncytio trophoblast, then
Cytotrophoblast.
cytotrophoblast, and then
Somatopleurer.
somatopleuric mesoderm. Okay, cool. We go here. So what's the top layer? Light blue.
Syncytial trophoblast.
Syncytio trophoblast. The cells here are cytotrophoblast, and this is the
Somatopleuric mesoderm.
somatopleuric mesoderm. But don't forget it's extraembryonic mesoderm, so that's why they wrote you here like that. That's just extraembryonic mesoderm. That's it. So we're trying to form villi, right? So if I go back to the structure here, right? So if I go back to this area, you can see that here we have some kind of protrusions. Why did I call it bushy, right? Because it is bushy. It will have lots and lots of branches just coming out like that. So these branches are called villi. So now we need to kind of refresh our memory of how do I form the villi, right? So how do I form the villi? So we're at this level, we're trying to form the villi. So how can I do so? So you can see that your syncytio trophoblast starts to protrude forward, right? It pushes forward towards the decidua, towards the decidua basalis, because this area right here, this is going to be decidua basalis, maternal side. So you can see that syncytio trophoblast pushes forward, it protrudes forwards until it kind of meets up with the decidua basalis. Take a look that inside of the structure, there is cytotrophoblast. So outer layer syncytio, inner layer is cytotrophoblast. How do we call the structure? This is called primary villus, right? So two of them, primary villi. So what do the primary villi consist of? Outer syncytio and inner cytotrophoblast. Later, you start to see that mesoderm wants to join the party. So it starts to push forward into the primary villus, making this villus not primary, but secondary. So what's the difference between primary villi and secondary villi?
The mesoderm.
The mesoderm, yes. If mesoderm is present, if this extraembryonic mesoderm is present, then it is going to be secondary villus. If anything, we have learned in embryology, in embryo 1, mesoderm can give you lots of things. Lots of things can be coming from the mesoderm, but it will always give you two things: connective tissue and blood vessels with blood inside. So this mesoderm, which is inside of the secondary villus, differentiates into blood vessels with blood and connective tissue. So how would you call this villus now? You'll call it
Tertiary.
tertiary. Okay, so this is known as the tertiary villus because it now possesses blood vessel and connective tissue. Just whose blood vessels are these?
Maternal.
No,
embryonic because it's
embryonic. This is syncytio, cyto, those embryonic tissue. So now these are the fetal blood vessels here. So in the late placenta, would you find all of them?
No.
Which ones are going to be the only ones in the late placenta?
The tertiary?
Tertiary, yeah, because they have the blood vessels, right? So, I mean, this is placenta. What's the purpose for it if I don't have a blood vessel? Because I need to exchange some things, right? So how do we call the space between the villi, right? So how would we call the space right here? This is called intervillus space. So this is the intervillus space. Fun fact, but intervillus space is actually here. It's this one, the trophoblastic lacuna. Intervillus space is this, the trophoblastic lacuna. So who is entering into the intervillus space? As you can see, maternal blood vessels, they empty into this intervillus space, filling them up with maternal blood. So good things diffuse this way, bad things diffuse this way. So probably there is a vein, there's going to be picking up all of the excretory materials of the embryo, so mom can get rid of it. Yeah. Okay, this part of the tertiary villus, so assume this is a tertiary villus because it's red, you cannot see it, but this is the tertiary villus, for example. This part, this is known as the stem villus, like stem of the tree, right? So this is the stem villus. This is anchoring villus because it's kind of attaching to the decidua. And all of these ones are called free or floating villi. Another name. Okay, so free or floating villi. So tertiary villus will have areas, right? So this is going to be the stem of the villus, of the tertiary villus, so stem villus. This is the anchoring villus, and these are the free. Okay. Hofbauer cells. These are APCs of a fetus. So on the fetal placental side, so somewhere here where the fetal villi are, we'll have Hofbauer cells, so macrophages just sitting there. Okay, so these are going to be the antigen-presenting cells there, so Hofbauer cells. Just some additional information, but I think we talked about all of it, except for this one, cotyledon. Cotyledon is a unit of placenta. Okay, so this is a unit of one placenta. So what is cotyledon? So you can see that this is tertiary villus. Do you see? This is one tertiary villus here at the bottom of this page. So how do you call the core? This is which kind of villus?
Stem villus.
Stem villus, right? What are these ones? Free or floating, and it's not visible, but let's imagine here it's attaching somewhere, so that's the anchoring villus. You can see that your tertiary villus is surrounded by some kind of decidual septa. So that's the maternal side here, right? Because that's the fetal side, because tertiary villus is coming from it. So that's the decidual septa. So the area between the decidual septa, this is known as the cotyledon. So if I go back here, where is my cotyledon? You're going to see the decidual will grow down like this, just a bit. It will grow down on this side, just a bit. So kind of surrounding your one tertiary villus, and outside of it, right? So between the decidual septa, this is the cotyledon. So on this picture, it's very weird because they kind of skipped the tertiary villi here. So there should be another tertiary villus, there should be another tertiary villus here as well, and another one here as well. So they're all right next to each other. Okay, how many cotyledons should you have? About 15 to 20. So there are about 15 to 20 huge tertiary villi like that. Here we're going to be talking about the placental barrier. So think about this way. If I go back, so could we name everything, right? So placental barrier, what's the most outer?
The anchoring.
Just do the layers. It's just the area here called anchoring villus, then stem villus, then free. But what about the layers? What's the top layer?
In situ.
In situ trophoblast is the first layer. Then
Cytotrophoblast.
cytotrophoblast. Then
Extra-embryonic mesoderm.
in the tertiary villus, we don't have extra-embryonic mesoderm.
Oh.
We have a blood vessel there.
Blood.
But you're forgetting that there's a basement membrane on which cytotrophoblasts are sitting. Then connective tissue, because this mesoderm differentiates into two things, connective tissue and blood vessel, then basement membrane of a blood vessel, and then endothelial cell. Okay, so that's the pathway of diffusion for the good things and the bad things. So if I...
So from the moment there is a blood vessel, there is endothelial cells, actually?
No, it's not an endothelial cell here, it's the intravillus space. It's filled with mom's blood here.
Okay.
On this side, in the villus, there is the fetal blood. And between them, we have different layers. So for example, if here I have like, I don't know, oxygen, and it wants to diffuse this way, right? So what does it pass through? First through this in situ, then cytotrophoblast, then basement membrane of it, then connective tissue, then base membrane of the fetal blood vessel, and then endothelial cell, and then now it's in the fetal blood. So that's just the layers that you're passing through.
The endothelial cells of the embryo.
Yes.
This is the last
because
layer.
this is tertiary villus, that's embryo structure.
Okay.
Okay, so those are the layers. I guess most important thing is that cytotrophoblast at the later stages will become discontinuous to make sure that it's much easier to pass for oxygen or like, I don't know, nutrients or something like this. So it becomes discontinuous to make sure that it's easier to pass. Okay, so that's what we're talking about, right? So that's the intravillus space that has maternal blood, and that's the area that we're passing through. Okay, so placenta is the endocrine organ. It makes some hormones. Just read it at home. Just read it at home which hormones it makes and what do they do. But it's the same thing. HCG, HCC. This is the somatomamotropin. It's basically placental lactogen. It kind of stimulates proliferation of the mom's breast. Okay, some additional things, but that's what it does. Relaxin softens the cervix and pelvic ligaments prior to delivery. And leptin, okay, so leptin is also going to be made there, especially during the last month of gestation. Basically, it makes sure that whatever mom eats goes to embryo. So it doesn't get stored in mom's, I don't know, fat, it goes directly to the embryo. That's what leptin does. Okay, finally, last slides, guys, of the semester. So congratulations. So we have here very interesting slides. So this is the pregnant uterus. Here we have some embryonic membrane attached without the embryo. They scraped it off. Here we have the myometrium, and that's the endometrium, right? But don't forget, in embryo, if you have embryo, you don't call it endometrium, you call it decidua. And this particular deciduum is actually deciduum basalis. So this is the maternal part of the placenta. If this is the deciduum basalis, then how would you call this one? What is right next to deciduum basalis?
The chorion.
Frondosum, the bushy one, the one that has the villi, right? So chorion frondosum, this is the chorion frondosum. This part is seeming that it is attached, but it's not. It should have been here. It should have been like that. But because they scraped off the embryo, it kind of collapsed this way. And this is actually deciduum capsularis. Okay, so this particular area, this is the deciduum capsularis, not entire thing, just this part. That's the endometrium here. If this is the deciduum capsularis, what is this one?
Cotyledon.
Cotyledon is, okay. is this. It's entire thing. Deciduo, which is located here, right? So somewhere here. That's where cotyledon will be, because it is one tertiary villus with whatever is outside. We just said that there was Deciduo capsularis, which is here. So it's on the opposite end of the
It's
cotyledon.
the Livia.
It's the leve. It's the chorion leve. So don't forget there are 3 maternal membranes, Deciduo basalis, Deciduo capsularis, and Deciduo parietalis. And there are 2 fetal membranes, chorion frondosum and chorion leve. If you see Deciduo basalis, right next to it is the chorion frondosum. If you see Deciduo capsularis, right next to it is the chorion leve. So if I go back, right, so this is Deciduo basalis. So whatever is right next to it, it has to be chorion frondosum. It has to be. So together, chorion frondosum and Deciduo basalis will make up which structure?
The chorion? No.
Chorion
Chorion.
frondosum. That's it.
The placenta.
Placenta, yes. Okay, so they will make up placenta. Okay, I'm scared to imagine what am I going to do with the rest of the groups, because these are 2 of the smartest groups and you guys are having difficulties with this. So what am I going to do with the rest of the groups?
Wish all the best for them.
Okay.
Okay.
Okay. Is it difficult?
It's the time, it's the hour.
I don't know, because I don't know. I personally, I don't think it's difficult. Just try to think it in 3D, okay? So if we go back here, so remember that this is the chorion frondosum. It's the fetal part. That's where the fetal villi are located, right? So this is the chorion frondosum here as well, all of this. Chorion frondosum has no need to be alone. It needs maternal blood. Where does maternal blood comes from? From Deciduo basalis. So this is going to be like the top layer here. That's the Deciduo basalis. Together, they are placenta. This is the placenta right here. Your embryo was inside of the endometrium. It was implanted in the endometrium. So of course, it is surrounded by a bit of endometrium as well. That's called Deciduo capsularis, right? And in the inner part there will be additional fetal membrane, which is called chorion leve. It's the smooth one. It doesn't have any villi. Why? Because there's no maternal blood. It's capsularis. It's just a capsule. It doesn't have much of the blood vessels, right? So all of the blood vessels are in the basalis. So of course, even if I had villi in the chorion leve, they will definitely degenerate. They will not be here. So that's why it's smooth. Do you see? It's very smooth. Okay, so let's do it again. This is the myometrium, Deciduo basalis. Right next to the Deciduo basalis, you will see that this is actually chorion frondosum. And then notice chorion frondosum is here, right? It's here, here, here. And then you see this area. And I told you that this is like a flap. It's kind of like this, because they scraped off the embryo. All of this area should have been this way. It should have been like this. So here should have been a cavity with the embryo in it, but there is no embryo because they scraped it off. So this area is a bit to the side in normal situation. So this is going to be the decidua. Just which one?
The liver.
Capsularis. Capsularis. Why? Because it's on the top of the embryo. So this is the embryo, it's on top of it like that. That's the Deciduo capsularis. And if you know this is the Deciduo capsularis, then what about this one here?
Chorion leve.
Chorion leve. So yeah. You can see a bit of the Deciduo that has nothing to do with embryo, right? You can see that's the embryo, it's the most far away from it, right? So this is going to be Decidua parietalis. There is this area here, which in our university, they call, I don't remember, but I think it's Decidua marginalis. You should check it. Okay, so if they still call it like that. Decidua marginalis is like a meeting point between all of them. Like if I go back, Decidua marginalis is here, this Because do you see that's the area where capsularis, basalis, and parietalis are all kind of meeting. So that's the deciduum marginalis right here. Okay, so take a look where we are. So what is this?
The decidua
Decidua
basalis.
basale.
This is deciduum basalis, yes. Which structures are these? Like this one. This one, sorry.
Tertiary villi.
Could villi be in the basalis? Are they in the basalis? They're
No.
attaching to the basalis, but they're not in it.
Maternal blood vessels.
Yes, these are maternal blood vessels. How do we call them?
Ah, this is the cotyledon.
Spiral capillary.
Okay, I go back. This is not cotyledon. More, you really want me to show you cotyledon? I cannot show you the cotyledon. You only see it in
But
the
it's not
in
me.
histo. You cannot see cotyledon. Forget cotyledon of the histo. Cotyledon is this entire thing. You need to see the entire tertiary villus. It's a big structure. The entire placenta has 15 to 20 of cotyledons. It's a big structure. You cannot see. This is microscopic thing of like, I don't know, nothing. So no, this is not a cotyledon. These are spiral arteries, super, super tiny arteries, which are inside of the decidua basalis, right? So like this one, these are also spiral arteries. These ones are actually uterine arteries, the one that I pointed here. Okay, so these ones are uterine arteries. You can see there's something pink inside them. This is actually glycogen. Okay, so glycogen is here. If this is the decidua basalis, then what is this?
The frondosum.
This is the chorion frondosum. So I will point at it in a sec. We're in the deciduum basalis, right? You can see that this is the uterine gland with the glycogen inside, and these are like branches of the spiral arteries. And you can see these dilated cells. How would you call them? In the deciduum basalis. They used to be called stromal cells, but now they're called
Decidual cells?
decidual cells. So these cells, the basophilic ones that you can see here, they are decidual cells. Yeah, so these ones are decidual cells. Now take a look where we are. This is the decidual basalis right here, right? So what about this part?
The frondosum.
Chorion liver?
Basalis frondosum. Basalis frondosum. Okay, so
Okay.
basalis frondosum. Okay, this one.
This is the liver.
This is the liver. And is this normal anatomical position?
No.
No.
Because it should have been here, right? Here should have been the embryonic cavity. If this is the liver, if this part is the liver, then what is this?
Capsularis.
Yes, this is the capsularis. Okay, so finally you guys are getting it. So this is the decidua capsularis right here. Yes. What about this little tiny point here?
The marginalis.
Marginalis. You need to check, okay, because I don't remember. I think it's called marginalis, but yeah. Now take a look where we are. We're in the chorion frondosum. We're inside of it. And this is still early because you can see all the different kind of weird looking branches. So for example, this is a fetal blood vessel. If you see fetal blood vessel, that means that how do you call this entire structure?
Tertiary villi?
Tertiary villus. This is a branch of the tertiary villus. We cannot see the entire thing, but this is going to be a branch of the tertiary villus. What about this one? Do you see any blood vessel in it?
No.
So?
Secondary?
Secondary villus. And what about this one?
Primary.
Primary villus. This is still early, guys, so that's why you see all of those things. What about this space?
Intravillus space.
Intravillus space. Who fills up this intravillus space?
Maternal blood.
Maternal blood?
Maternal blood, very good. Maternal blood. We magnify a bit more. So what can you tell me? Which villus are you looking at?
Tertiary.
Tertiary, because you can see that blood vessels are starting to develop. Blood vessels are here, then what is all of this? How would you call it? What is all of this?
Syncytial.
Since
It's inside where the blood vessels are.
cytotrophoblast.
Cytotrophoblast is outside. This is cytotrophoblast cells.
Okay.
A mesoderm.
The
Not
mesoderm.
mesoderm, but okay, guys, if this will make me go back, but we had their mesoderm, but in the tertiary villus, it gave us two things: blood vessels and
The connective tissue.
But we're in the embryo, so how is this connective tissue actually called?
Mesenchyme.
Mesenchyme. So you say that this is actually mesenchyme. Okay, so this is going to be mesenchyme. So in the tertiary villus, the mesoderm gives you two things. It gives you blood vessels and connective tissue. Just don't forget this is embryo, so it's actually mesenchyme. Outside of it, these cells are cytotrophoblast. You see them individual cells with the plasma membrane, one nucleus per cell. Cool. On the top, you see weird looking cells. There's just lots of nuclei, but where are the cell borders? Impossible to see. So these cells are syncytiotrophoblasts, yes. So that's the layers, right? So syncytio, cytotrophoblast, then connective tissue or mesenchyme, then the blood vessels. Okay. So many more, but okay, more inbar. You should be very happy because we can see the cotyledon. We can see this entire thing is the cotyledon. Everything that you can see here, it's one cotyledon, this entire thing here. So what do we need to know? Here I kind of put it there for you already. So first of all, three villi are everything that you can see here. All of those weird looking things, those are the three villi. Okay, all of them. The three of floating villi. Deciduo basalis is here, right? So this is the deciduo basalis. This is the stem villus of the tertiary, huge one, right? Stem villus is this one. And you can see that this is the deciduo basalis. So there's a part where it kind of attaches, right? Like here and this area here. So this is the anchoring villi, right? And this area right here, this is the chorionic plate. Chorionic plate is basically syncytio, cyto, and whatever mesoderm has given you. Like here we can see it's already blood, right? So blood vessels is already there. So we magnify and what do we see? This is decidua. How do I know that this is decidua? Because I can see decidual cells. So this is a branch of maternal blood vessel, probably spiral artery. Sorry, I'm just looking at my, do you guys know this book? Do you know this book? It's for the step one. It's the one that you need, you know, for your simile. And do you know, here I'm looking at the structure, and it for some reason reminded me what we're talking about right now. So that's why I got a bit distracted. I was like, is this part? And then I realized, no, that's actually gallbladder. And I was like, never mind, okay. But yeah, sorry. So just to explain you why am I looking away. But yeah, histology follows me everywhere in life. But yeah. So these are the decidual cells right here. Because I can see the decidual cells, I can tell you that's the decidua. So how would you call all of these weird little tiny branches?
Spiral.
Spiral arteries are in the decidua. We're on the other side of the decidua.
Three villi.
Three villi, very good. These are the three of floating villi. Notice how many of them are here, right? So this is the three of floating villi. How would you call this one?
Stem villi?
Anchoring.
No, anchoring.
Why anchoring? Because you can see it's attaching.
Okay.
It's attaching to the deciduo. This is the anchoring villus. And what about this one then?
Stem.
Stem villus. That's the stem. You would still call it stem. Okay. So if you see something like this and I point here, what would you say?
Stem.
Stem. This one? also stem, right? So also stem, because I cannot see the decidual, I cannot see anything like that, so I will still say that this is stem. Inside of the stem villus, you can see that there's some kind of blood vessels, so definitely that tells you that this villus is actually tertiary because it possesses blood vessels. So these blood vessels here, they are actually fetal blood vessels. So all of them here, fetal blood vessels, right? And the rest of them here are free or floating villi. So we magnify, and you can see that majority of this free or floating villi possess blood vessels. So they are all branches of the tertiary villus, right? So let's do the layers. Please try to do the layers. Very difficult, but okay. The top cells are definitely
Syncytial trophoblast.
syncytial trophoblast. What about here, for example?
Cytotrophoblast.
Yes. Do you see that they're discontinuous? They're not as we saw before, because this is a late placenta, right? We can see so many blood everywhere. It's a late placenta. The first placenta was early. It was quite continuous cytotrophoblast. Here they're discontinuous, like this is as well, right? Blood vessels, right? And this is the mesenchyme, so embryonic connective tissue. That's it. So, gratulanok, and we are done.
Thank you so much.
Thank you,
Thank
Julia.
you. It was fun, guys.
Thank
So,
you.